Organization student my organization student
Good morning, sir.
Good morning.
Sorry, sir. Sorry, we have live. My shipment is coming from Japan. That's why I'm middle of the work. Sorry.
Yeah, no issue, sir. Now okay?
It's okay, sir. Yeah.
Yeah, your voice is clear and our video is a bit. I mean, can you put just camera setting? Yeah, I can see you properly. Yeah, perfect. Yeah, perfect. Glad to see you, sir. This is Dr. Kumar, the director of Vishnu College of Pharmacy.
Thank you, sir. Thank you, sir.
Yeah. So welcome to this webinar.
Yeah, sir.
You're okay, we'll start with the proceedings.
Yeah. I will share my presentations.
So Nagarjuna Reddy, everything set? Should we start the proceedings session?
Can I
Good
share
morning, one and all.
uh,
I, R. Sanjan of third year, second year M Pharmacy, Regulatory Affairs Department, heartily welcome to you all to the first day of 3-day national webinar on current rapid growth on vaccine manufacturing and regulations in India, in association with Indian Pharmaceutical Association Education Division. Now we'll have an AV of Sri Vishnu College of Pharmacy. Now, I cordially invite our beloved director, Sir Dr. Kumar Nimani, to address the participants.
So, good morning to each one of you. Myself, Director Sri Vishnu College of Pharmacy, on behalf of management, staff, and students of the college, welcome you to this national webinar on current rapid growth and vaccine manufacturing and regulations in India. I believe this topic is very appropriate at this juncture, and it has its potential long in the future. And I congratulate the Dr. Nagarjuna Reddy and his team for selecting this team and organizing wonderful speakers. The testimony of this program, the success of this program is one-way measure in terms of the registrations. At this juncture, we are having close to 1,000 registrations, which speaks the quality of the speakers of today's as well as the upcoming two sessions. So at this note, I really thank Dr. Mahesh Kumar Garu for accepting our request and are ready to take on a very important aspect, that is the overview of preclinical toxicology studies on lab animals for production and regulatory requirement, especially the vaccines. So as many of you know that the CCMB is in news because it is the organization who did the initial studies for 2-deoxyglucose, the efficacy studies, especially showing the antiviral activity for the 2DG. And there onwards, it went for other studies and got approved from the DCGI. So apart from that, CCMB is known for many, and Dr. Mehshar and many of our students are know about that your institute and are all aspiring to come to your institute and do PhD. So I'm sure with your today's lecture, they will even fall in love with your institute and get motivated and understand the current concepts, especially in developing vaccines and those regulations, and make their future career in this field or in the pharma field in abroad. So with this juncture, I like to thank each one of you and suggest each one of you to active participation, interact with the speaker, and also participate in the upcoming webinars. This is a webinar series we are conducting. We just completed a series from the Quality Assurance Department. Now is from the Regulatory Affairs Department. And we do have another 4 departments which are upcoming national webinars, a similar series like this. So your participation is welcome and hope this will be an enriching experience for your future careers. So once again, thanks Mahesh Garu for accepting our request and over to you.
Thank you, sir, for your valuable words. Now, it's my privilege to introduce our today's guest speaker, Dr. M. Gerald Mahesh Kumar, Senior Principal Scientist at Center for Cellular and Molecular Biology, Hyderabad. He completed his bachelor and master's from Veterinary Animal Science University, Chennai. He completed his PhD from School of Pharmaceutical Sciences, Kumamoto University, Japan. After completion of PhD, he worked as lab animal veterinarian for the period of 4 years at National Institute of Immunology, New Delhi. In the year of 2007, he was awarded as JALA's International Young Scientist from Japanese Laboratory Animal Science Association, Japan. In 2007, he achieved JSPS Ronpaku Fellowship to get a graduate program at School of Pharmaceutical Science, Kumamoto University, Japan. He completed his graduated program in the year 2009. In the year of 2012, he honored with Laboratory Animal Bursary Award for the purpose to get 3 months training on animal imaging techniques at University of Stuttgart, Germany. He completed 2 research projects by ICMR and Department of Biotechnology. He published 132 papers in peer-reviewed international journals. Now, today's speaker, Dr. Gerald Mahesh Kumar, will give us an overview of preclinical toxicology studies on lab animals for production and regulatory requirements. Now, I request our speaker to address the participants.
Good morning, everyone. Can I share my presentation?
Yes, sir.
Yes, sir.
Desktop, that is there, my desktop. It's opening now.
Yes, it's opening, sir.
Yes, it's open.
Yes.
Okay. Good morning, everyone.
Yeah. Morning.
Okay. Yeah, thank you for nice introductions. I'm working for the past 20 years in this laboratory animal field. Basically, our institutes working on basic research activity, and recently we are mostly involving with the COVID-related experiments. Since I'm starting with these presentations, people are working on this pharma lines, and what are the scopes in this in CCMB and CCMB related laboratory? Because the people are working on B Farm or M Farm. The level of B Farm, we have the opportunity, the summer training program. This every year, this month of April, it will come for advertisement. Those who are pursuing B Farm 3rd year, that you can go with them for application program for 2 months training program. They will give them, they will allocate some kind of lab. They can go and get a training. And another one is dissertation program. That is the 4th year of B Farm students are M.Com student, they want 6 months or 1 year, depending upon their curriculum, they can accommodate them. Such kind of opportunities are available. All the CSR labs, since I am working in CSR lab, many CSR labs are there. We have this, our parent organization, Indian Institute of Chemical Technology, very adjacent to the CCMB. They are also offering similar kind of program. I request all the BCom and M.Com students, they can utilize this opportunity to use for such kind of improve your career, definitely. Okay, coming to our today presentations.
సార్ ఎవరో ఒకరు ఇక్కడ కూర్చుంటారా?
Is it visible my screen?
ఇది సెంట్రల్ ఇది చూసుకోండి. అవసరం అయితే ఒకసారి పిలవండి.
Visible, sir.
Visible, okay. Yeah, this is content of my presentation, and specifically you are voting on vaccine with a regulatory requirement. Then let's say I am working on animals. I am an animal point of view. What are the things are required for regulatory purposes? Either this production of manufacturing, either in the level of R&D level. What kind of basic criteria required for this animal experiment in vivo? They will do many kind of in vitro studies. Once the in vitro studies and in vitro studies are carried out, then they are replicating their in vitro experiment to this preclinical study. If you have the in vitro data, if you have the preclinical data, either the drug or either in formulations or any kind of vaccine, that need to go for this clinical studies. That is a basic requirement for development of new R&D. If they are, that is one type of the requirement. Another one type of the requirement, this regulatory requirement, regularly the large number of animals used for every batch release of any kind of test substances in the formulations or in these new drugs coming to the market, and any vaccine, this every batch release that you need to be tested certain kind of test criteria, that kind of pharmacological studies, certain toxicological study, that's regulatory requirement that record large number of animals. Basically, the animals used for different purposes and different kind of experiment that we are going to discuss for that. And you can see prior to that, we can read what are the title of my presentation, introductions and history and breakthrough in animal research. Why such kind of animal model is necessary? If the drug development, what kind of animal model is necessary, that I am going to explain briefly, and what type of animal model, where the sources animal models are available, and coming to the conclusions. You can see them throughout the globe. This is not a 80 to 100 million. If already we have crossed in 2010, more than 100 millions of animal. Every year we are utilizing the animal for different species, from starting from zebrafish to non-human primates. The major animal model, what the purpose they are using, pharmaceutical companies they are using, and contract research laboratories. Specifically, there's many contract research laboratories. India have more than 48 GLP laboratory, good laboratory practice laboratory, and they are extensively they are using for any kind of those preclinical studies they are doing under this umbrella. And again, many medical school, many basic research institutions, they are extensively using animals. And what kind of their research they are doing? Basic research activity also, and also applied research activity also, educational purposes also. Majorly we are using extensively animal. What I am highlighting few of the breakthrough using animals. You can see them. This is first the major discovery of insulin. It was how they discovered. You can see them, Dr. Frederick and Dr. Colip and Press. These three are medical students. Now, medical student, they are going for master degree. They are giving some kind of project, small project. At the time, very familiar with them pancreas. Not much detail about the pancreas, but some kind of secretions are involving with the metabolic activity. Nothing know about that. What is the essential functions of the pancreas and its secretions? What the student they have done, they want to do in depth of a study of this pancreas. What they have done this, their boss given the 10 dogs for their experiment. They have 5 dogs, they remove the pancreas, and they are finding the very similar symptom of diabetes symptom. Nowadays, what the diabetes they have named there, and what are the clinical signs are the typical symptom they have clearly observed in the dog, cramping, frequent urinations, sweating, all kind of symptoms are very clearly visible, the prominent. Then they are very surprised. This is the, then they, what happened, that they have the pancreas, they have to homogenate and they have isolate. The best is very good eminent person for filtrations or such kind of technique in the lab, and he did the filtrations of the pancreatic secretions. Again they re-inject the animal, re-inject the dog, again the dog come back to the normal stages. These are all a very surprise finding. This is the major achievement through this using the animal. Through that it was developed. This is a novel insulin they have discovered. Because nowadays the recombinant insulins are available because 2000 till 2000 year, we are mainly using for isolated insulin from different kind of animals and secreted, purified, that animal only we have used for this diabetic purposes. But now we have the recombinant technology will be developed. Now we are using for recombinant insulin for all kind of purposes. This, unless until early science, we are not aware that what is insulin. That kind of the discovery can be happen without use of animal is very tough. Similarly, the polio vaccines also similarly they have passaged of this earlier day. The polio encephalomyelitis virus is very, very critical disease for all over the world. The patient will die. We don't know. We ask the viruses, but the various very much invasive in the spinal cord and nervous system. They have homogenate the spinal cord, isolate the viruses. The viral they bring into the culture level. The culture level, from the culture only they are developed, inactivated them, the Salk, he was developed the vaccines or polio vaccines, this inactivated viruses. Such kind of technology they have developed without using animal, without finding in the animal, they cannot be achieved. And again, many medical procedure, kidney transplantation, heart valve transplantation, such kind of the things are they are not. Still, people are the many big surgeons also they are going first get the training in these animals, especially the big. They are using for many of the heart surgeons. They are going trying. You can see the many cath lab in animals all over the world will be there. I don't know. Maybe you are aware of that. Even Hyderabad, we have the cath lab, specifically the animal specific cath lab in Palamur Bio. They have this many doctors. They are coming for training. And very typical lab, you can animal will be anesthetized and they have to implant them different kind of implants specifically in the aorta and coronary artery to dilate the blood vessels. Such kind of procedures they have adopted in the animal novel stent, drug-eluting stent. That are all they are testing in the animal. Such kind of things cannot be coming out to the market unless until you get the what kind of output, what kind of reactive changes in the animals. You can see the many breakthroughs is there, but we are coming at the why is necessary, whether it is required. The animals are required for that. If you are developing any, why you are developing, but many new drugs are coming up. Is it necessary for that? We're all asking such kind of question. Definitely, that's required because we are not know what kind of the changes happen in the microorganism, specifically the bacteria, viruses. So viruses have this behaving different property, different kind of mutations. You can see just an example. This COVID viruses, how it is mutating. It's entering into the different kind of the disease person. You can see that the Delta variant, any kind of variants is changing their proteins, changing their amino acids, the pathogenicity and the spread of infections is severity it happens. We can't predict them in futures what kind of genetic modification will be occur in this virus. Again, the bacteria also we don't know this nowadays. We have many end of antibiotic and we are following not a proper regimen. Automatically what happens, the antibacterial resistance will develop. It will be leads to many kind of bacterial resistance and the development of the new antibiotic is mandatory for that. For such kind of situation, if you have the viruses behaving mutations and different kind of approaches and antibacterial resistance happens, such kind of critical situation, you need to strengthen your R&D that require a proper usage of animals. That's why this is proper regulatory requirement to guide them, regulate them, number of the usage of the animal. You can see them in India level, OECD, the OECD Organization of Economic Cooperation and Development all over the world. This will be 64 organizations, 64 countries are under that OECD. India in 2012, it was joined under the OECD. This OECD it will be recognized the GLP lab all over the India. Those who want to test the animal all over the globe, they can send them, they can do under the GLP tested lab that can be accepted, that can be marketed into their company. That is one of the added advantage for using such kind of umbrella under that OECD or EPA, Environmental Protection Agency. In India, they have the regulatory link called Central Insecticide Board under this DCGA. That are all the regulatory body that will be regulating them. Once the drug is developed under the preclinical and clinical stages, that need to the specific accreditations lab that to get the marketing of this drug. Okay, you can see them coming to the research purposes, how much animals are using. You can see them, the pharmaceutical research is extensively using in the animal. That's mean the drug developmental research purposes, only the extensively they are using around mainly the pharma and vaccines. 84% they are using them. Remaining 16% only they are using for other purposes, either educational purposes or defense purposes or food additive purposes they are using. What they are doing them, that's very important. You can ask them, what is that they are doing in the pharmacological test? This is basically they are doing any kind of the drug or vaccine need to be tested in animal studies, 3 different categories. One is they have to study for this potency. What is the potency? Potency means whether the drug, whether the vaccines are effective or not. That is a potency. And second is efficacy, and how long it is, how much time. That's efficacy. Efficacy also in form of the effect of the drug. Efficacy. Another one is safety. This is a potency, efficacy, and safety. These three parameters you need to thoroughly check unless until do with many kind of preclinical studies. That is very important. Very kind of preclinical study at the level of either R&D, at the level of it will be the mass production stage. That you have to look for them. You have to what kind of potency study, what kind of efficacy study, what kind of toxicity study that have been defined by the each and every regulatory requirement. India, the DCGA Indian regulations, they are very clearly defining what kind of potency study to. If you have the vaccine, suppose I am telling you vaccine, the COVID vaccine, many people they have manufactured. In India also they have the Biodegradable, they have the R&D level they have developed. But what kind of test they first they have? They have to study the efficacy. What kind of efficacy study? The first efficacy study, you look for them immunogenicity study. What is immunogenicity? Immunogenicity means whether the vaccine is eliciting immune response or not. How do you assess them? Whether it is really the immune response? First, you have to assess them what kind of animal model you can use. Because this COVID is, the hamster is a very appropriate model. In otherwise, we have to go for this. Normally, the mice have the ACE receptor will be the deficiency mice. In human have ACE receptor is there. The viral COVID virus is entering through the ACE receptor. But in mice they have deficiency of ACE. But if you want to go with using for animal, you have to go over ACE transgenic mice. The mice will be expressing ACE receptor in their lung pneumocytes, then that animal is most suitable. But this ACE transgenic mice, CCP, we have ACE transgenic mice we brought from Jackson Laboratory, and even though we have developed ACE transgenic mice for specifically these purposes. What do you mean that you need them? You have to, for the first thing, if you are going to test them your vaccine, specifically potency and efficacy study, you have to first select the model. What model you have to use? If you are COVID, if you are vaccine, you have to go with a specific model. If you are testing the vaccine model, then you can check the immunogenicity. How they are testing the immunogenicity. You can listen how you can test the immunogenicity. You have to inject them the vaccine formula. Suppose inactivated vaccine, the Bharat Biotech, they are acting inactivated vaccine in a different levels. This inactivation at the different steps. Pentapropiolactone. Normally, the industry they use different kind of secret formulations. Anywhere else, you can. The PPL is the common inactivation, commonly using inactivation of the viruses. Okay. If you are once inactivation, the virus is injecting to this specific model. Suppose the hamster they are injecting, and they have to bleed them in different dates. Like this human, what you are doing that you are injecting, you are checking the levels of antibody titer. That is the things called as immunogenicity. How the immune response achieve in the animal, how they are checking, they have to check them. This titer, antibody titer, whether you have to check them. Otherwise, same way you can check with them different test called neutralization test. Whether really the antibody is neutralizing the viral particle or not. If you are neutralizing, how much the viral particles are, how much IC50, how much incubatory concentration, 50% of the viral particle died with how much concentration of the inactivated formation of formulation. Such kind of things need to be calculation. Such kind of in vitro studies and in vivo studies that are all necessary to such kind of any kind of formulations, any kind of drugs. That's a major purpose using R&D purposes. That means for efficacy and potency. Then you are coming for safety. Safety, you are going for toxicological study, many kind of toxicological study, different kind of parameters required. That means you can start from acute study. Acute study means you are doing with a single dose. Dosing with a single dose, you can keep observation on 21 days animal. Whether the animal is died, whether the animal is showing any kind of clinical signs or not. If these animals are not showing signs, this animal have no toxicity. You can straight away, you can go them and you can tell them, suppose you are administering the lower level to higher level, you can start with them 300 mg, start with 1000 mg, then 2000 mg, 5000 mg, every dose level up and down procedure. You are going with the toxicity with up and down procedure. You are going with a different dose regimen, whether the animal is showing any kind of toxicity or not. If the animal is showing the toxicity, you can record them. This is the level of toxicity. Such kind of parameter, going with a single dose toxicity, that is called acute. Then again go for multiple dose toxicity. That means you are going continuously administering for 28 days, and you can check for not only clinical signs, you can check for go for clinical parameters like hematology, biochemistry, and different kind of systemic organ. Whether the organ have any kind of histopathological observation, you have to look for any kind of micromorphological changes, whether have any kind of necrosis, any kind of inflammation in the liver or kidney, all the internal organ thoroughly check whether really the organ showing any kind of toxicity. If they are drug or vaccine, they have not showing any kind of toxicity, then you can declare them. This is the levels of no toxicity. You are saying no observable effect of toxicity level, not observe such levels of dose. Thus, your such kind of toxicity study you need to record for any kind of production or manufacturing of the drug at the level requirement is very, very essential. Okay. Can come into this. This is the set of parameters. If you are going for any kind of the drug under development, you need first a pharmacological. You can go for any kind of big company. Just an example, the Sai Life Science is there in Hyderabad. Again, GVK is there. Like and Syngene, Biogon, like many multinational company, they are developing a models in vitro and in vivo setup that will basic requirement do it for any kind of either under the R&D level or it's going to the manufacturing level. It's going to the manufacturing level is very, very important to study the pharmacological and toxicological study. Okay. And what kind of the study? This is just a flow charts how what kind of pharmacological study and toxicology study they are going. Pharmacology means you have needed separate oncological experiment, pain and inflammation, metabolic disorder, respiratory disease, and neuropathology. This is the module. If you are developing any kind of drug, such kind of department will be there. You have to analyze them whether have any kind of effect of drug, any kind of toxicity of drug. That means this a basic principle is efficacy, safety, and potency that need to be tested in all kind of the system. That means you have to look for the pain and inflammatory level, metabolic level, respiratory level, neuropharmacology level. You have to look for them whether this is effective or not. Such kind of pharmacological test you need to do. That is the main things required for R&D level or manufacturing level. You can in vitro toxicity. In vitro toxicity, what they will do? They will do in vitro toxicity. They will be Ames test, chromosomal aberration test. What are the named? I am naming the test. What they are doing them? They will be collecting them specifically the bacterial mutations happen or not. If the mutation happen, that will be have any kind of toxicity level. They will be assessing them again. That is called genotoxicity. That means if it will be have the drugs are potentially to generate possibility of carcinogen. Like this have declaration, you need a mutagenicity test. That means the in vitro pharmacology they are doing some chromosomal aberration test, Ames tests that will be there, that will indicate them whether have any kind of mutation happen or you have any kind of toxicity happen, that will be assessed by the in vitro test. Again, you can go for DMPK, direct metabolism pharmacology. If you are administering the drug, how much the concentration of the drug is achieved in the circulation, how much optimal concentration reach, what level, at how long it will be existing. That means you are bleeding the animal from the 0 point to 72 hours continuous bleeding of the animal in different time point and checking them where the dose formulation whether reached, how it is reached, such kind of things. Then only you know that whether really your drugs is bypassing this blood-brain barrier system. If you are suppose you are targeting the neurotargetic drugs, any kind of nervous diseases, whether your drug is reaching to this, passing to the blood-brain barrier, such kind of things you record. You need there such kind of DMPK studies. That means you have to make them fine homogenate. You have to assess them the secretions, whether your formulations has reached in the secretion or not. That kind of setup is very essential, required for all kind of regulatory purposes. Okay, and you can see them. Then for such kind of setup, what kind of animal model they are going to use? Some of them they will use for spontaneous model. Some of them they have to experimentally induce them. What is you can suppose you want to do for experimental for diabetes? Diabetes or experimental inductions are available. You can for chemically induced, or either you have to go for diet induced. Like a many model they can be created that can be support to study the drug efficacy and safety of the drug, efficacy and potency of the drug. Okay. And chemical induced model, you can see them, this is a pain inflammation. Just I am going with the system-wise system. If you are going for metabolic disorder, you can go with them different kind of hybrid induced DAS model to study the inflammatory conditions. Again, if you are going for pain and inflammations, you have the many chemicals inducing pain into this animal. They have to first assess them. They have how you have to induce them. Either the capsaicin is a chemical. If you inducing the hyperalgesia, I mean it's creating more pain to the animal, and you have to assess them. How do you assess them? Nowadays you may be aware that your lab have many kind of pain assessing equipments are available, but you have to induce them by such kind of capsaicin or front complete adjuvant or carrageenan. These are all the chemical. You can induce them the pain to the animal, and you can test them level of pain or inflammation. And like a heart plate, what do you have to test the animal how often they are taking out the paw. That means there's a time point that will be recorded, and it will be assessed them how immediate response is coming. That means how it is making immediate pain to this animal. If you are administering any drug and the animal is slowly taking up the paws, that means the takeover time is increased. That means this drug have effect on the pain conditions. Such kind of things you have to assess them. Many kind of equipments are available, nalgiometer and heart plates and many kind of the equipments nowadays commonly available, but you have to create the model, and this is very, very important. We're using different kind of chemicals. You have to create the model and to assess them what are the activity of your drugs. Again, you can see them even metabolic disorder also. You can see the OGTT, oral glucose tolerance test. You have to first create them diabetic model. How to diabetic you have to create? Suppose you are chemical inducing them. Chemical induce you have to go with the streptozotocin or alaxan. You are creating the models, either acute model or chronic model. What is acute model? What is chronic model? Acute model in single dose, and single dose, the higher level of dose, you are injecting them either streptozotocin or alaxan, and immediately the destruction of the beta cells, animal is going to the high glucose level. Such a model is called such a model for glucose disorders. And again, the chronic model, you are multiple doses, you are administering slow levels, and to achieve them to impair glucose impairment. Such kind of things. How do you assess them? Whether this really your drug is working or not? You are going to do with them oral glucose tolerance test, oral insulin tolerance, oral insulin tolerance. Whether that means whether you are administering the glucose level and you are administering the drug, whether challenging them, whether it is working or not. Such kind of things you need to test them. 1 minute, sir. I have one. Yeah, that means very important. You have to what kind of tests you are assessing the levels of glucose. That means you have to normally once the model achieves glucose level, you can check with the glucose level, the model is achieved. Then again you have to go with the challenge with the glucose. Challenge with the glucose. That is very important, whether your formulation is working or not. Glucose tolerance test. And again, insulin tolerance test. Whether what is the levels of insulin is increasing or decreasing. That kind of test you can do, assess them, whether the formulation have a good efficacy or potency, that can be understood. Coming to this respiratory model, like many models are there, that means the area is different, the pain and inflammation, metabolic disorder, respiratory diseases, but every system you need to develop a model, and according to the model you have to test them efficacy and potency. If the drugs are entering into the good efficacy and potency, then you can go for them only clinical safety and toxicity. But if the drug is safety also, efficacy also, then you can drug straight away. You will go for all kind of preclinical parameter, all kind of basic assessment parameter. Then you have to register under them for the clinical studies. The clinical study, if the drug is going to register for clinical studies, there is the DCGA or OECD. They have EPA. What else to be their organization? They will ask for clear target. Whether have you cleared for all the safety parameter? Whether have you cleared for all the potency study, in vitro study or in vivo study has happened or not? Such kind of things is very mandatory. Whether they will assess them, whether the preclinically the drug is safety and potency, that you can definitely replicate to the animal, replicate to the human model in the clinical experimentation. That is very mandatory for all the drugs discovery programs. This is a respiratory model. Respiratory models also, many models are LPS induced model is a very good model. Nowadays, many people using, and fluomycin induced model. These are also they are using. This you do not have such kind of animal models are available for any kind of vaccine. Normally, the COVID viruses extremely infecting to the lung infections. You do not have a proper model with because the AC transgenic mice are not available all over the world. But many of the pharma, what they have done, they have induced them LPS. They have LPS, whether this they administer through this respiratory tract. How they are administering? With the intranasal administration of this LPS, and it will be spreading to the entire lung. That will be automatically. That will be the acute reactive changes in the lung that will be creating the high neutropenia will happen. Normally, the condition of the COVID also similar kind of phenomena is happen, and then they are doing testing of different kind of the drug, many kind of anti-inflammatory drug, whether they are working the levels of the inflammatory conditions of lung has reduced or not. Such kind of studies need to be done. Such emergency situation, you have to create the model that is very mandatory. You can see them coming to the model for cardiovascular diseases. You can see them cardiovascular. Normally, the basic mechanism for all the cardiovascular diseases is atherosclerosis. What is atherosclerosis? Inflammations in the major arteries. But how it happens if you have any kind of high bad diet for administering, high bad diet 40% bad or 60% bad with cholesterol level. That's very important. The cholesterol level, how much percentage you are adding them. Either 1% or 1.2%. If you are adding the diet, the diet will be administering for more than 2 months. Definitely, the animal will develop such kind of atherosclerotic condition. That means the high fatty level condition. The animal have always with the high total cholesterol level, LDL, HDL, all the things will be happen in this similar phenomena occurring in case of high lipidemic patient. What happens is the high lipidemic animal have highly prone for all kind of cardiovascular disorder, like a human, many kind of cardiovascular arrest, cardiovascular ischemia. Ischemia means lack of blood supply to the heart. If the blood vessels have accumulated with a plaque condition, the fatty plaques in the atherosclerotic plaque in this coronary artery of heart, what happens automatically? The blood supply to the heart will be coming down. That lead to this narrowing the lumen, lead to the cardiac ischemia. That means the lack of blood supply to the heart, automatically the cardiac function will be collapsed. That will be the cardiac arrest will be happen. Similarly, the functions of the blood supply to the brain will be coming down, automatically the brain ischemia will develop, automatically lead to the paralysis. Such kind of things are happening, stroke, such kind of things are happening. But if you want to create such kind of model, that is very important. Either you have to go with the surgical type. Many surgical models are available. This is the occluder, or if you are using for a large animal, U-shaped occluder and ring-shaped occluder, balloon inflation, such kind of things you need to completely do with them. A big surgical setup of normally they are doing with the big and heart. They have the such similar kind of human surgery they need to do with them. You have to animally or put it into the ventilator. Ventilator, you have to give them the artificial respirations. Again, it will go for heart lung machines. The heart and lung function completely shifted to this machine. Then you can open the thoracic cavity and you can separate the coronary artery and you can place them this clamp, this U-shaped occluder or ring-shaped occluder, that you have to concise the flow of the blood to the cardiac myocardium. Automatically the animal is developing the ischemia. The similar kind of model will be developed to the human. This also will be done. Now, many animal models mouse models are available that you need a very fine surgical equipment and microscopic units and record. Even we are CCMP, we are doing many myocardial infarction model. And after myocardial infarction, what we are testing them different kind of stem cell and progenitor cells they are bringing us, and whether the regeneration happening or not, how it's regenerating, how much the capacity of the cardiac muscle regenerating happen, and at the molecular level we are studying for them. Then for such a kind of situation, how do you do them? You have to animal put it in the ventilator, opening up the third and fifth thoracic vertebrae and expose them heart coronary artery. You are getting similar steps. You can see them in the images. The animals are in this ventilator, and again they made the ligation in the coronary artery of left side ventricles. Automatically the last you can see them this after immediate suture, you can develop them completely the infarction, the lack of blood supply to this left ventricle. The animal will be still alive for certain time point, more than a week, and we can test them whether what kind of regenerating capacity happen. That can be studied possible. That means that you have to test them any kind of drugs or any kind of stem cell or vaccine. You need to create the model is very mandatory as per the regulatory requirement. That means then only they have the regulatory people will be accept. But how to assess them whether they have really the inboxes happen or not? You have to go with the specific staining, NPT staining. Otherwise, you have to go with them. If you are animal, you don't sacrifice. You have to check for levels of enzymes of cardiac troponin and creatine kinases. That will be give them very clear indication. If the levels are raised up, then definitely the myocardium has damaged happen. If you want to sacrifice the animal, you can go with the NPT staining. The very clear, the black color staining happen around in the myocardium, specifically the left ventricle region. That will indicate them whether they will be you have achieved them the infarction model for this mouse. And similar kind of studies are available. You have to check for this cardiac pressure, and you have to look out for any kind of variations in that mean arterial blood pressures, diastolic blood pressure that can be measured will be possible. And again, if you are sacrificing and going and measuring them, different kind of quantifications also can be done possible with the immunostaining. Okay. And again, very simple stuff, even your kind of pharmacology people, you can do them in your lab also. What we are doing them, you have to create them trans-aortic constriction. You cannot open them thoracic cavity, and you can exactly there below the neck, you can make the incision to look for where is the aortic arch is coming. You can very clearly visible for aortic arch, and you can ligate them with the ligating with a very superficial threads, and then you can look for them flow of the blood will be come down, and automatically what happen, you can see them cardiac vagale will develop. This enlargement of the heart develop very progressive, because you cannot type them here only one side. You are ligating them the aortic arches, and the progression of the from the heart will be coming to the blood will be slowly come down, but slowly the pathological changes achieved in certain time, but within a month the heart will be slowly the pulsing. You can in your laboratory condition, if you are M4 people, you can test for them. If you have the compounds, have them specifically working on them. On the myocardium or inflammatory, severe inflammatory condition will come down, such kind of formulation, you can create them trans-aortic constriction that you need not any kind of the cardiac machine, you need not any kind of ventilator not required. You can simple, yes, good surgical setup, a clean environment that will be efficient to do with the mice or rat with the trans-aortic constriction, and you test your formulations. And apart from that, if you have not to have such kind of setups are not available, you can go with the chemical induced model, doxorubicin, isoproterenol. There are many published literatures are available. You create, mimic them, the myocardial infarction model. Then you can test with your drugs whether this drug have potency or not. Okay, again, there's many transgenic models are available. Apo E protein, this apolipoprotein E. This is one of the transporter of protein, transporter of lipid. That kind of that apolipoprotein knockout models are available. It's a very good model. This always the animal will be showing high level of cholesterol level and high incidence of high risk.
Bhaiparaja, could you please some noise disturbance is coming?
Yes, sir.
Yeah, Mahesh sir, please go ahead.
Yeah. That's mean the system-wise different kind of animal models are available, but you need to list out whether this requirement of regulatory what they are expecting from you. And if you are going for any kind of new model drugs, you are registering for your drug, what is the basic things you have to test it into the animal? That's a preclinical level, what kind of things, potency or efficacy, safety, what kind of things you have to study, that need to list out, and you have to go with the different kind of parameters. That means if you are new drugs, and the basic parameter, whether the drug is reaching to your system first, whether they're reaching the system, what level it reaches second, and then whether it is reaching to this specific system. That means specifically the body have blood-brain system cannot be reached possible. Some of the drug cannot be reached to the brain. Such kind of things you have to look for them, whether the your drug you have achieved to the brain. That means this is the basic parameters. You have your drugs are reaching certain concentration. Then you have to what kind of effect is there. Then you can what kind of safety is there. These are all the basic list of the requirement for the regulatory for any kind of drug discovery program. And if in case of the marketing the drug, if you are already the drugs are in the market, the drug only going for safety study. It cannot go for again go for potency, again it won't go for all the profiles, only for safety study. Safety study also it will not go for all the toxicity. Normally the toxicity study involved with acute study, subacute study, and 90-day chronic toxicity study. 90-day study again it will be go for carcinogenicity study. Carcinogenicity study you can do with them 2 years. That such kind of things will be at the levels of the drug registering purposes. But the drug under already in the market, already in the production stage, already coming in the market, it cannot go for such kind of toxicity study. But you need to do certain kind of parameters to regularly do with them, like acute toxicity study. Pyrogen test. What is mean pyrogen test? If you are formulation, you are injecting to the animal, the temperature is elevated or not. Such a pyrogen test. Such a small kind of test, whether the drug is potency, small levels they can go for, not many kind of tests. That means major concentration, the drug already in the market, go with the safety profile. safety profile only they will do them. Whether they have any kind of contaminations are there or not, that they cannot be tested again for that. But only if you are going for a new formulation, if you have already, you can see them paracetamol with aciclovir acid. Then if you want to change them some other drug, adding with them paracetamol, aciclovir, along with I want to add one more formulation. Fine. If you are doing the formulation, you need certain kind of test need to do. But you need not go for all the basic parameters of basic test record, not required for them. But certain kind of formations, certain kind of basic safety profile studies will be required that you have to look out for them in the animal studies. One minute. You can see them. This is the cardiovascular parameter. I told you the Apo E model is very, very good model for cardiovascular disease. You can see them the plaque development, how the big development of plaques to obstruct the flow of the blood. That will be a very good model for a cardiovascular ischemia. You can see them very, very good model. And if such a model, you can see the blood pressure, how much rate is there, pulse, how much is there, heart rate, how much is there. But such kind of things cannot be created with the normal model. What you will do them, you have to go with them hybrid diet. Hybrid diet, you can administering them the hybrid diet, either the 45% of the hybrid diet or 60% of the hybrid diet, and the duration, either you can go for the 2 months or 4 months, whether the levels of achieved were desired parameter. That means you are looking out for them whether the total cholesterol level is reaching more than 500. Then you can start with your experiment. Such kind of things, you have to plan your experiment for cardiovascular studies. These are all our experiments. This is a basic parameter. If you are going for R&D drug discovery program, you need to thoroughly experiment them, all kind of parameter. You can see them, what kind of these levels of expression of different kind of protein associated disorders. That means if you have inflammatory conditions, what are the parameters that are elevated? The CD4 elevated, CD8 elevated, or how much level of elevated, such kind of things, whether your drugs, you are injecting them, whether the CD4 levels are coming down, the inflammatory cytokines, pro-inflammatory cytokines are increased, or anti-inflammatory cytokines are increased. Such kind of parameters at the molecular level that will be required for any kind of drug recovery. This is a very in-depth studies that is called pathway. Then again, the gene expression profile. Many kind of the gene expression profile you have to study for them. Whether a set of genes involving a specific pathway, that if you are going for one drug, if you are registering them, if your drug is acting which pathway, it is which pathway it is blocking. That means what is the pathway? The gene, what is the arresting of the gene further coming down the levels of expression. That means if you are apolipoprotein is the major protein involving the transporter of the protein, transporter of the lipids. But if the levels of apolipid change, automatically the deteriorations of all the lipid transfer will be changed. You check them the related parameter of apolipoprotein pathways, whether your drug is altering the expressions of associated genes. If you are altering them, definitely if the drug is working on them. That means the drug, if you are going to prove them all kind of molecular levels and in vitro level and again preclinical level, then only the drug is more suitable for coming to the clinical profiles. Okay, that is very, very important. So again, this stroke model, how do you create them? Stroke model, basically the many people they are using with them surgical model. And you can see them, this is the diagram very clearly explained. You can go with them either 2 vessel or 4 vessel model. That's 2 vessel you are tying them, or 4 vessel tying, either temporary or permanent. That means normally the cerebral artery supplying to the brain. You are ligating them a common current, either your common carotid artery you are ligating or internal cerebral artery you are ligating. You have to ligate them, automatically the flow of the blood will be coming down to the brain, automatically the ischemia, the lack of blood supply to the brain is specific portion. If you are common carotid artery ligating, the whole of the brain is showing the complete ischemia. Then after 24 hours or 48 hours, animal will showing the paralytic symptoms. Such kind of things, you have to look for them, whether specific region you want to develop them ischemia, then you can go with the 2 vessel disease, and again you can go with them, you can go with only the cerebral artery, either right or left, or either right carotid artery or left carotid artery. This is the way we have to ligating the flow of the blood to the exit portion of the blood, exit portion of the brain need to be coming down to the develop a ischemic condition to associated disorder of stroke, similar to the human being. You can see them. This I have done many colleges, many pharmacy colleges. We have went for workshop. We have the ligating them common carotid artery, and we have observed them. This is the within 3-4 hours, the movement of eyelids will be defective. movement of eyelids will be defective. Even these are all not a major surgical equipment, not the things are not required. Lab level, you can do them. Even I have explained many kind of pharmacy college, even Kovaraji, Rangaraji College, and CMR colleges, many colleges, I have done such kind of workshops. You have to ligating them, common carotid artery, and then you have to see them very clear. Within 1 or 2 hours, you can see the movement of eyelids, and next day, definitely the animal have this right or left limbs paralytic can be visible. But such kind of model created, afterward you can test with your formulation whether the drug is potency or not can be possible. And again, thromboembolic model, also similar model. You have to make them the clots, the blood clot. You have to make them in your pierces, small pierces, and then again injecting into the carotid artery, and it will be depositing into the brain different region. That will be the blood clot development, and that will be going to the embolic development to the specific region of the brain. That will be creating the ischemia develop, and again associated symptom will be developed. Such model can also be possible. These are one of the surgical models. Okay, and like many models, depending upon your system, can be created is possible. Okay, and this is the bone disorder model. This also we have created many kind of pharmacy colleges. What will do if you have any kind of implant, specific implant like a calcium or magnesium specific implants that can be potential regenerating capacity, that you have to create them. You have to create the surgical model. How do you create the surgical model? You have either the rat or rabbit. You can develop them. The rabbit femur bone. You have to select them from the femur bone. You have to make them drilling of this bone. Yeah, bone driller. You can use them, and from this driller you have to implant your specific implant, and you can live for this animal for a month. And whether have the what is kind of the regenerating capacity happen, what is the bone mineral density, what is the bone mineral composition, what is the calcium level, what's the phosphorus level, such kind of thing you can measure them, that you can indicate them whether the level of your regenerating capacity of your implants. Nowadays very popular, many kind of implants are developed, and with the drug coating implants are also now the developed, many people are using them. But such kind of machine, you have to create the model, either the femur bone, you have to create them, you have to create the bone defect, you have to create them. You can see them. This is a surgery. After sacrificing the animal, also you can live animal also, you are checking them whether how the implants are set up in the bone. And this is mainly the machines called DEXA. The DEXA machine will give exactly the bone mineral density, how much bone mineral composition, that will be very clearly indicate, and calcium phosphorus ratio, and that will give them very clear indication of the regenerating capacity of your implants. And again, if you are after sacrificing animal, you can go with them specific immunosaining that will indicate them levels of osteoporosis. You can see them in these images wherever the red cells are there. Those are bone forming cells. This is the implanted site. The implanted site, you can find them wherever the red color indication for this is the osteoblast and osteoclast proliferations and initiation of growth. That means that's a bone, your implant has acting on them, initiating to them, the cells differentiating into the bone forming cells. These are all to identify them whether your implants is working or not. By the immunostaining is called acid phosphatase. Okay. Like many transgenic models are available throughout the world for to study them different kind of the drug. Okay. And I am not going for the transgenic knockout model. It's again very high chapter, and I'll come with them. But the trend, just I brief them what is transgenic and knockout. Not transgenic means the specific genes inserting into the mouse genome, and knockout is specific gene knocking out them from the genome. Then this is a specific set of procedure that we are regularly doing for the creation of transgenic knockout point. This is a very tough procedure, but the success rate of the knockout is very less, 10% will be there. But you have to record the assembly of microinjection setup record. You can see the microinjection setup. I'll show you. You can see them. You have to superovulate the animal. The first thing you have to superovulation. What is superovulation? If you are injecting with the FSH and LH hormone, automatically the ovulation will be higher. The more number of ovum will be released. Ovulation means ovum release. The more number of ovum will be released, and then automatically the animal, we are putting the mating to this male fluent fertile male. The next day morning, that means 0.5 days. Next day morning, we are take out the animal, sacrifice the animal, you are collecting the oviduct. In the oviduct, you are cut open, you can see them, such kind of beautiful embryo, you can see them. In this embryo, you can see them, this embryo top small dot-like structure is there, that is called male pronucleus. You can very clearly see the inside one big nucleus is there, above that one small nucleus is there, that is called male pronucleus. In this, under the microscope, this is a small dot, you have to inject your gene of interest, that is either 5 microliter or 10 microliter, you can inject them. After that, you have to transfer this embryo into the pseudo-pregnant animal. How do you develop the pseudo-pregnant? Mean animal, what we are doing them, the animals are in cyclic condition. You are checking the animal whether the animal in estrus or not. The animals are estrus. The animal estrus animal, you are put it into the vasectomized animal. What is vasectomized? Vasectomized means vas difference will be removed. The vas difference is very, very important for the transfer of sperm to coming ejaculation. If the vas differences are removed, the sperms are not transported, animal is impotent. What happens is that the vasectomized animal, you are put in mating for the proven fertile, that means cyclic animal. That means animals are under the estrus animal, we are putting the mating with the vasectomized animal. The next day, the vasectomized animals, next day the pseudo-pregnant animal feel the pregnancy. Animal feel pregnant, that means pseudo-pregnant. That animal only we are doing embryo transfer, and then we are checking for the delivery. If the checking, and again you have to screen them whether your transgenes is there or not, whether your gene is knocked out or not. Such kind of molecular tools, you have to isolate them DNA, and from the DNA you can go with a specific gene, whether inserted or not. That you have to go with the PCR and 7 hybrid. Then you can check them, proven but them that knockout is developed or transgenic developed. Then again you have to characterize the mice. Once your mice are developed, then you can go for characterization. The characterization is very, very important. You look for them whether they are achieved or if you are Apo E. That means whether you have the cholesterol level achieved or not. Such kind of thing. The functional studies that will evaluate them. Your model is developed. Then it will go for testing your drugs. Okay. Such a, maybe in brief discussion, we have checked many kind of system, many kind of system. You have to develop a model. That's very, very important. The pharmacology, if you are pharma, many kind of pharma company, they have the R&D setup for different kind of unit. That will be neuropharmacology. You need a system-wise pharmacology will be there to in-depth study the in vitro and in vivo assays. Okay. And coming to the source of animal, India have few source of animals are available. You know that Biodegradable is a Biodegradable is a tie-up with Taconic. The Taconic is the major breeders for Europe. And again, Charles River also have the Indian agencies are there, but Charles River not directly coming here. Jackson also, they are not directly coming to the India. But what people they are, the Charles River, they are already tie up with Hylasco in Hyderabad. The Hylasco, the breeding nucleus they are supplying to the Hylasco. Hylasco is the major distributor of Charles River animal to the all over the India. That means recently the many people are coming about here, but since we do not have a specific repository in India, majorly we are depending upon this repository from all over the world. This is the source of animals. Coming to the conclusions, several medical advances discussed would provide evidence benefit to the human for use of animals in research, but there is no substitute for living system to study the interaction among tissue or organ. Animals are the best surrogate because of their similarity to human in stature and function of their organ and biological system. Thank you.
Thank you, sir, for your valuable session.
Yeah. Bhaviraj, should I take ahead? Give the
Sir, participant has some queries. Can we proceed, sir?
yeah, please.
Participants, if you have any queries or questionnaires, you can ask the sir.
If there is no question, please come to CCME with your educational tour. CCME is always accepting the students visit. And in the group of 40 or 50 people, you can come and see them. CCMB, IACT, NIN, like many institutions are here, many pharma institutes are there, many pharma companies are there. You can come with the industrial tour and look for what kind of setup we have.
Sir, thank you, sir. Thank you for your valuable input regarding the animal models and everything. Sir, I want to propose a vote of thanks, sir.
Okay.
Sir, respected Dr. Gerald Mahesh Kumar, sir, we are very happy and we are very happy that we had a very fruitful session from you, sir. And today, actually, we conducted this national webinar on current rapid growth on manufacturing and regulations. And in that, we are very happy that you gave a very insightful webinar on these models for regulatory requirements and breakthrough in animal models. We discussed, we got insight on the breakthrough in animal models, why we are using the animal models, and what are the types of animal models, and what are the sources. And that's all from my side, sir. And we are in future we are ready to have such interactions frequently, sir. Thank you, sir.
Thank you, sir.
Sir, so now our director, sir, will have a concluding session, sir. Sir, please.
So thank you, Mahesh sir. It's a wonderful session. I think it's a delight for us. I was so happy to see that you covered the major topics, especially how research happens in a research institute like CCMB, and typically in industry also. The topics that you covered, especially the specializations in the rank order that you put, like oncology, pain, inflammation, metabolic, respiratory, and neurological disorders, is interestingly my research expertise too. I have done that in my industry. For the past, I have done for about 20 years industry, has done exactly those models. Most of the models you mentioned as we have done some maybe thousands of molecules screening, maybe in lakhs of animals. It's a delight for me to talk, and I'm a little bit regretting that I don't know how I didn't miss you in interaction so far.
Thank you, sir.
We'll make this, it's a good choice that you accepted, and we'll continue our interaction with you. And as you mentioned that we welcome you for a workshop. We are also planning to conduct workshops in pharmacology. In the PG syllabus, whatever you taught today, I teach in the classes here, exactly the whole syllabus in a regular fashion. So I think you are very valuable for us. And the options that you mentioned, like our students can come and visit your labs and also do some projects. Yes, we are looking for such collaborative interaction. We did set up a center for preclinical research excellence, a small animal house, a kind of industry setup, not a bit above the academic standard. And we dream to establish all these models except cancer, because cancer, as you know, is very costly affair for academic research purpose. Otherwise, my heart and mind is for research. In cancer, I work on immune therapy, and drugs are being sold to the multinationals, and they are in advanced stage of screening. If they get, they're like, I'm hoping at least one of them will get approved and come to the market. So with this note, I thank from SVCP management and as well as the faculty and the students, and looking forward for the fruitful interaction. And you should visit us also.
Thank you, sir. Thank you.
And we will welcome you for that. Yes, on your suitable convenient date. So thanks.
Thank you, sir. Thank you.
Sir, thank you, sir. Sir, from our It's my pleasure to thank Dr. M. Jiral Mahesh Kumar Garu on behalf of management, director and principal, sir. Once again, sir, in spite of your busy schedule, you have accepted this webinar program, and also you have enlightened our students with a great knowledge, sir. Thank you, sir. Thank you.
Thank you. Thank you, sir.
With this, I will conclude this session, sir.
Thank you, sir.
So, all the participants, I will provide the link for tomorrow due to technical issues. We have changed our webinar link, and so I will be forwarded this link to all the groups and also to all your emails. Thank you for your kind cooperation.